According to Guinness et al., 1983, the varying morphologies of thylakoid membranes inside of bean chloroplasts were observed closely using electron microscopy. More specifically, the study was interested in what would occur to the proteins that are critical for photosynthesis under intentional heating conditions at increasing temperatures. While they found that the rate of the electron transport chain was altered due to heat stress, they also found that this might have occurred because of the disruption in the chlorophyll protein complexes that constitute both photosystem 1 and 2, as well as the separation that occurs to the non-bilayer lipids. The structural organization that takes place in the thylakoids after being exposed to larger amounts of heat coincides with the separation of the non-bilayer lipids and the change in the rate of electron transfer between the photosystems. And another piece of evidence from another study shows that according to Semenova in 2004, structural changes were observed within the thylakoid membranes of chloroplasts derived from peas with a change in temperature. Because the overall thylakoid system changed its morphology at increasing temperatures, this could mean that certain proteins were also changing conformational shape, potentially indicating a change in function. The protein's functions within the thylakoid membrane, where the electron transport chain occurs, could thus be affected by a change in temperature, especially when heated. And lastly, for our experiment, we were interested, after seeing all this other data from these two studies, how heat stress as well as a lower change in temperature might affect the rate of the electron transport chain in thylakoid membranes of spinach. We decided to use TCP/AP as a molecule that would serve as an electron acceptor in its oxidized form and would thus interrupt the rest of the electron transport chain by preventing plastiquinone from receiving the electron passed on to it from the photosystem 2, and then it would no longer be able to further pass it down as it normally would throughout the electron transport chain. And because TCP/IP has a blue color, we would be able to see the change in absorbance at 580 nanometers once recorded.